And I'm happy to introduce our next speaker, who is a returning star. It's the first time, though, that he's here in person. So let's all welcome David Sinclair to the stage.
Okay, Morten, you're going to give my talk today. Yeah, and I'll ask the questions. Well, thank you for having me here. It's been 10 years, and you finally got me here. Actually, if you were here last year, Alex made me promise, looking straight down this camera from the other end, that I would be here this year. And so this is actually one of the few conferences, perhaps the first that I've been to in person since 2020. And boy, is it a great meeting. This is the one meeting that I didn't want to miss. So thank you, Morten. Thank you, Danielle and Alex. You're always inspiring with your energy and love of this topic. All right, so I don't have a lot of time. In fact, I'll put a timer on here so I don't have to get dragged off the stage, because I can talk all day about science. It's very exciting. All right, so it's good to see old friends and lots of new faces as well. I love how there's a new generation of scientists coming into this field. And even just 10 years ago, as Morten and Alex and Danielle can tell you, this field really was nowhere. In fact, when I was starting to work on aging medicines or longevity medicines, it was widely regarded as a crazy thing to be doing. So it's really, I got to say, really emotional to be up here today. Thank you. But you're not here to listen to me talk about my emotions. You're here to see some data. And so I'm happy to share some new data with you. I'm not going to talk about a few things that have been happening. I think that's, can only talk about one main topic. So I'm not going to talk about the NAD work, unfortunately. I'll give you a quick update. NAD boosters, the main one that's being developed into a drug at Metro Biotech, has been in humans for 3 years, is showing efficacy. We've published a couple of papers on that already, and others have shown similar results of lowering cholesterol, lowering blood pressure, and improved what looks like improved endurance, like we saw in the mice. We're seeing something of a signal in humans. So I'm not going to talk about that. And my name's all over the internet with NAD. I don't sell NAD supplements, by the way, but a lot of people think I do. But what I want to talk about and focus on today is what is a new topic and an old topic. It started my career, and it may very well end in my career. If hopefully there isn't an end to my career except at my funeral. I want to keep doing this. So those of you who were around 30 years ago will remember that the epigenetics of longevity started around the mid-1990s, largely based on work in yeast cells, which I was very fortunate, in fact, unbelievably lucky to land in a lab, the lab of Lenny Guarente at MIT. Brian Kennedy, who's here, was the postdoc who was, graduate student, I forget, he was already so mature. He taught me how to wrangle yeast cells. This is an old yeast cell in yellow. I've stained it with bromophenol blue. It's acidic. And Brian taught me how to study yeast cells, and those were some golden years. We had 4 years where collectively as a group, I won't go into exactly who made the discoveries, but Brian initiated the project, Matt Kaberlein came in, Shin Imai, who discovered NAD and the sirtuin connection. It was a golden age, and many of the scientists in that group went on to become famous scientists in their own right. What we discovered in those days, and I'll give Brian credit for discovering the SIR442 mutation, which led to the sirtuin field as we know it now. The sirtuin field was interesting because when we first discovered it, or when Brian came up with this finding of SIR442, it made no sense. We were thinking we're going to find a gene involved in DNA repair or a gene involved in antioxidant activity. Instead, out came this silent inflammation regulator number 4. What the heck? That makes no sense, nothing to do with aging, right? But over the next 6 to 12 months, we started to figure out that there's probably something to do with silencing and aging and controlling of gene expression. And we proposed, or Brian proposed, that there's an age locus, which turned out we discovered is the unstable ribosomal DNA. But all of this is now history. What's more important is, what did that teach us about our own aging? And that's what I want to tell you today. So sirtuins, if you don't know, are epigenetic regulators. They deacylate proteins, the lysines on proteins, including histones. And in doing so, they can regulate many proteins, including the compaction of chromatin. And the reason I mentioned, I said that the sirtuin field is interesting, is that we got distracted as a field in the 2000s. Now, we did some good work as a field. We found that the environment controls the sirtuins, the amount of glucose, the amount of temperature, amino acids. We linked a lot of signaling and environmental inputs to the sirtuin activity. But what we really didn't focus on for a long time, and we should have, is the epigenetic role of sirtuins. We were more interested in mammalian sirtuins controlling p53 and NF-kappa B, and isn't that really cool? It all makes sense. But really what bugged me as a scientist was, what about the epigenome? What is going on in mammals there? And so what we've come up with, and I'm just going to jump to the chase before I show you data, is what we're calling, and others have called, the information theory of aging. So what I'm trying to get across with this theory is the following, that aging is not just a breakdown of our bodies. Right? We're not just cars that wear out and then we die, and we try to fix little bits and live longer. What this theory says is that there is encoded in every cell information that keeps the cell young, and as what this diagram is showing, is that there's what we believe now to be a backup copy of that youthful information in every cell. I used to give the analogy of a scratched compact disc that got a bit old, and I didn't want to explain what a CD was to young people every time. So now the analogy is more like rebooting the software in an old computer. And this is based in concept on the work of Claude Shannon in the 1940s and 50s in the information theory of communication, where he proposed that there may be a backup, or we should make a backup copy of every signal of information that we send out, radio waves, and now the internet. And now we've got the TCP/IP protocol of the internet. And what I think we've got now is an understanding that there's a TCP/IP protocol of biology, that there's a backup of youthful information encoded somewhere, and we think we know where, though that'll maybe next year we'll talk about it, where that information is stored. And we can access this using increasingly easy ways. So I'm going to talk about 2 ways to reset a cell epigenetically and get back the youthful information. One is genetic means, and we'll talk about Yamanaka factors to do that. And you heard Sharon last night tell you that we're already in monkeys and improving vision using Yamanaka factors to rejuvenate the eye. And we're also going to, at the end of my talk about perhaps much cheaper ways than gene therapy to reverse age. This is an old slide, but I love it. This is by Jan Vai, who you heard this morning, and Yusin Su and others, published in Cell. This was a summary of a paper that Philip Oberdorfer, a postdoc in my lab, published in Cell back in 2008. And what we did was we said, okay, what's going on in yeast cells is the movement of sirtuins to the RDNA where there's genomic instability, and that distracts the sirtuins from what they should be doing, which is silencing genes, such as the mating site loci, which control sterility. And that's a hallmark of old cells, sterility. And we thought, well, you know, we're not yeast cells. We're not going to necessarily just get sterile from the movement of sirtuins, but maybe in principle it's the same, that genomic instability distracts the sirtuins from their main job of controlling gene expression, and perhaps they don't come back to where they originated in full. And over time, it's like a tennis match where you lose the balls and you have to go collect them. So this paper was the result of many years of work of Philip and our colleagues, showing that that principle is true in mammals, that the sirtuins in the nucleus, we focused on SIRT1, Vera Gorbanova, I think, as you know, has done a great work on SIRT6. moving to DNA breaks and helping repair. And the movement may actually mess up gene expression over time. And as you can see here, it's also a very active process. This movement of sirtuins to repair broken DNA requires signals such as ATM and H2AX. So this is not just an accident, this is an active mechanism. Now you might say, well, why would cells do that? Why would cells mess up their epigenetic software over time? Why not just put it all back where it came from? And we don't know, but my theory or my idea is that this is a very adaptive, helpful response. So when cells get damaged, there's a response. Epigenetic factors like the sirtuins and probably many others, PARPs, go to help with the damage. There's a signal, they temporarily go there. And that's coupled with transcription so that genes that are involved in repair and metabolic changes in response to damage coordinately get expressed. So in this system, we saw that SIRT1 leaves to go where it's needed to repair DNA, and these genes that were once silenced by the sirtuins come on temporarily, such as DNA repair and metabolic changes, like more glycolytic. And then, if all goes well, then the sirtuins come back, the DNA repair proteins get shut down because you don't need them around as much, and metabolism goes back to normal, all great. But probably what's happened is that we haven't been selected to live beyond, about 50, 60 years. And so the system works pretty well for young cells and young organisms, but not so well when we're old. And that's known as antagonistic pleiotropy, as you know, and it's perfectly consistent with that. So what are we doing now? Well, we wanted to test this theory, not just look at what's happening, but test whether this is perhaps a cause of aging, not just something that happens during aging. And to do that, we built a mouse that I've been talking about for 13 years, so I'll go quickly because you're probably tired of this mouse. Fortunately, we published it finally after 13 years. We got it out there in January. The ICE mouse stands for inducible changes to the epigenome. What we did was we mimicked genomic instability by cutting the chromosome rarely in a mammalian cell or a mouse. And we showed after many years that this does not cause an overt accumulation of mutations. In fact, we could not find mutations that were caused by the cutting of this enzyme called PPO1. And also, one of the criticisms of the paper by some is that this is probably causing cell death because it's causing DNA damage. Well, actually, we designed this system to be very, very mild. These cells do not die. We see no evidence of apoptosis in the cells or in mice when we turn it on. We typically turn it on for 3 weeks. And I call it chromosome tickling because it's not a lot of DNA damage. The cells, in fact, when you grow them in a dish, they don't even slow down in their cell cycle, and they don't show on a Western blot a DNA damage response. Right? P53 does not become phosphorylated even when you turn on this cutting enzyme. Cells can deal with it really well, but it's enough to tickle them so that the sirtuins get distracted. And 3 weeks later, we've got a change in the epigenome, but not a change in the genome. That's very important, right? We know that mutations can help drive aging, but we wanted to separate damage and changes to the genome from changes to the epigenome. And using this system, I believe that we've done that. So now we can ask the question, do changes to the epigenome have anything to do with aging as well? And the answer that we came up with was probably, because the mouse that was treated when it was young for 3 weeks with this system did have epigenetic changes that we can see that are very similar, if not identical, to normal aging. Transcriptomics, we did proteomics, mapped most of the changes across the histones. And the mouse you can see on the left there is the one that was treated 10 months beforehand. And then we think we initiated a domino effect of change to the epigenome. And the kind of things that we saw in that mouse were very similar to mouse aging. So we can also look at very precise changes to how the DNA is folded and modified. That's what this slide is showing, but I'm not going to go into details. But these red lines show DNA contacts. And after the treatment with the cutting enzyme and everything's repaired, if you do the genome, you say there's no difference. DNA is the same. But if you look at the folding of the DNA with these red loops here, big differences, okay? And so we think, or at least we're testing the hypothesis, that these changes to how DNA is arranged in cells is a major driving of disease in mammals. And you can read this, so I'm not going to go through each one, but we looked at a lot of different tissues with our collaborators. There were 64 scientists, 15 different labs around the world. Rafa de Carbo's lab helped us greatly. among others who are here. We looked at the clock with Vadim and Steve Horvath. We're looking at bones, we're looking at eyes, we're looking at skin, muscle, and every time we gave tissues to another lab, they came back and said, yeah, this is aging, okay? Including the epigenetic DNA methylation clock, which was invented during the study. So just to finish up, now we're going to ask the question, if you can give something to an animal, can you take it away? If you can give aging, can you reverse it? And so Wencheng Liu, who now is at MIT as a postdoc, was a very brave student. He took on the project. It was circa 2014. And his project was to find the backup copy, if there is one, of youthful epigenetic information. And he almost gave up because we were trying genes that were involved in embryonic development. Of course, we tried all 4 Yamanaka factors. We kept getting tumorigenesis. We didn't want to cause tumorigenesis. We wanted youth, but no further. We didn't want to go back to age 0. And he almost gave up, and he was, I think he wouldn't mind if I told you that he was to the point of tears in my office. And my job as a professor now is not to do the work, but just to make sure that my students don't quit. And this was one of those moments where Wencheng was ready to quit. And I said, is there one more experiment that we can try? Because my gut tells me after all these years that we're onto something. I just don't know what yet. And he did a killer experiment that worked. Instead of putting the 4 Yamanaka factors in, which is what Yamanaka himself won the Nobel Prize for in 2012, and also I should credit the Belmonte lab, who had shown in 2016, showed that if you pulse the expression of 4 Yamanaka factors in a mouse, a progeroid mouse, you can extend their lifespan. So we knew that that was true. But 4 Yamanaka factors is overtly toxic to human and mouse cells. Okay, unless you pulse it, you have to be very careful not to cause tumorigenesis or death. But what Wencheng discovered, to his delight, was that using the 3 Yamanaka factors and leaving off MYC, we could drive cells back 75% in age, but no further. There was a barrier to getting back to 0. And this was our breakthrough. We maintained the cell's identity. Actually, we regained the cell's identity, but we didn't go so far as to erase it. And so he did a really great experiment, which was to now go into an animal. This is in Jigang He's lab, where he does this model of nerve damage. Now, we chose the eye, not because it was easy, but because it was hard. And also because gene therapies are already approved for the eye. And we thought, if this works, hey, we could try it in humans. And so what he did was to pinch the optic nerve of a mouse, or a few mice, and have a look. If you reverse the age of those cells, what happens? And we know that if you damage an old nerve, it doesn't grow back. But if you damage a young nerve, it can grow back. And so this is the control, the no treatment optic nerve. On the right is the eye, and the left is the brain. And you can see, without the orange dye, a lot of the nerves are dead after this is after 6 weeks. The optic nerve is largely dead. Otherwise, it would be bright orange. But after turning on the 3 Yamanaka factors, that was sufficient to not just protect the cells from dying, and we turned this on after the damage, so this is not just pre-treatment. We can do it 24 hours, 48 hours after. But these nerves also started growing back to the brain, and in fact, we left them for 16 weeks, and many of them went back to the brain. This was unprecedented. But this isn't aging, this is wound repair. So we did glaucoma with Bruce Cassander and his team, and glaucoma worked. Mice got better at seeing. And then I said to Bruce, can you try aging? And most scientists who've never heard of studying aging think this is a crazy thing. But I pushed him and I said, just try it. I've got a good feeling about this again. And so he did old mice. And this is some figures from the Nature paper that came out in 2020. So we use adeno-associated virus to deliver 3 of the Yamanaka factors. It's doxycycline inducible. And you'll have heard Sharon last night saying the treatment that she's going to try in humans will be doxycycline inducible. So you can get your vision back, hopefully, and then repeat every few years. In these mice, we can do that. And we treated them for 4 weeks and asked, can blind mice be made to see again? Before I get to that result, we looked at the DNA methylation in these cells, and it was largely restored versus the control. This is now genome-wide methylation. We worked with Morgan Levine and Vadim, Steve, to look at methylation age, and it was younger in the treated cells. in vivo. This was the figure that blew my mind. I did not expect this. This figure shows that there truly is a backup copy of youthful information in vivo. Why? Because if there was no memory of youthful gene expression, those dots, which is individual genes, would be flatlined across horizontal. But the fact that they're along the diagonal means that the cell knows which gene to change the expression and in what direction and by how much. That's 3 bits of information in there. And as I said, where that information is stored, we're actively looking. The mice got their vision back. We now know we can do this multiple times. And one of the surprises, nice surprises, was that the changes to the DNA methylome were very similar to the injured eyes that we had done previously. So what I believe is that aging can be driven by epigenetic changes that can be accelerated by a few insults to the cell that we know of. One is DNA damage or double-stranded breaks. The other is crushing cells, which is what I showed you. Vadim's lab has now shown that stresses to the body, even pregnancy, can change biological age. So I think we're starting to see that aging is a manifestation of cells freaking out in response to damage, including DNA breaks, and that is a driver of aging. I want to show this again in case you missed Sharon's talk. OSK treatment of non-human primates improves their vision. And so this is a model of stroke to the back of the eye in a monkey. And the results, which are very clear, is that these are electrical signals at the back of the eye. And the treatment without the Yamanaka factors, those monkeys are blind. They have a lot of nerve damage when you look at them. But after we treat the monkeys with the OSK, a lot of the neurons survive and the electrical signals are largely restored. Now, we don't know if these monkeys can see again because that's hard to measure, but there is no doubt that you can restore the function of a monkey's eye by reversing the age of those animals. And so we're excited about testing this in people in, I won't give a timeline, but it's certainly within a short time frame. As soon as we get enough virus and approval to do human studies, we will initiate those. We're doing, just briefly, because I know we're out of time, we're doing work on the brain now. So we're just about to submit a manuscript. This is Xiao Tian's work out of Vero Gorbanova's lab, who's in my lab now. We grow organoids. We can age the organoids out to age 70 within a week. We can give them Alzheimer's disease, model of Alzheimer's. And then we can reverse the age of those. And using this little system here, we can see that the electrical signals of these organoids, cerebral organoids, come back again. We can express OSK now in very specific regions of a mouse's brain, including neurons of the hippocampus. And what we can see is that those mice that are treated by age reversal of the hippocampus get their ability to learn again. And we have some hint that even lost memories are recovered in those mice. So what I've said is that we think we can reverse the age of cells epigenetically. We can use Yamanaka factors. It's going to be expensive, even if it works. Gene therapy is not going to be available to everyone on the planet. So what I'd like to see is something for $10 that we can all take to rejuvenate. So the final slide that I have is some advances that we and others are making around the world to reprogram cells using chemicals. And the field's going super fast. We put out a paper with Vadim's lab just a few weeks ago in aging. And this is using an assay that looks at the integrity of the nuclear pore complex, which is, of course, important for aging. And these are some of the chemical structures of chemical cocktails that reverse aging in cells. And we will be testing this in vivo shortly. These are the controls on the left here, these two. And then using transcriptional clocks, which Vadim's lab has developed, you can see that each of these 6 chemical cocktails reverses transcriptional age of these cells. And these are just different clocks that they're using. So I believe that we will be able to reverse the age of cells, at least epigenetically, using chemicals. I believe that we'll be able to one day do this for $10. And we'll be able to do it multiple times throughout our lifespan. So just to finish up, we've got evidence that when you damage a cell physically, perhaps even metabolically, there will be changes to the epigenome, and that this is reversible through, I think, what's one of the most remarkable discoveries in a while, the cells have a memory somewhere of epigenetic usefulness that we can tap into. So lots of people, as you can see, helped, and these are just some of the highlights. On the left are the people in the lab who I mentioned along the way. We've been helped by many people who are here at this conference. I apologize if I didn't mention you. I'm very grateful to everybody who makes this possible, and the funding agencies as well. And some of the people here I've highlighted, and John Sedevy's been helpful as well. He's here today, and he helped us do some of the work with understanding where the sirtuins are moving to and if they're involved in activation of viruses, or at least retroviruses, which they seem to be. So I'll stop now and I'll take a question or two if we can. Thank you.
Thank you so much, David. We have time for, I think, 1 or 2 quick questions.
Hi, my name is Andrea Olsen. I just recently graduated high school, and I'm continuing my university education now in Caltech. And I have a question for me and really the student community. So there's a lot of research going on right now, but what is going to be happening in 20 years? What should we focus on? What topics? Thank you.
Okay. Andrea, that's a good question. So basically, what should you be studying to stay ahead of the field? Well, I see a radical transformation. It's almost hard to see 5 years ahead now. We used to be able to, it used to be slow, but now things are going so fast, my head's spinning. I'm also an optimist, and I'm not even able to predict where we're going. The merger of AI with chemical screening, with understanding biology is happening right now. Alex is, years ahead of most people as usual. But this is happening. I've been visiting various countries who are leading the way in this, and this. We're going to be able to do 1 billion experiments that would normally take, well, what would take normally thousands of years we can do in a day. That's probably a better way of doing it. So what are the areas? Well, I think our ability to control biology is what we're seeing finally. It's the culmination of understanding what are the major hallmarks of aging that happened in the 2000s, 2010. We pretty much agree on what the major problems are that we need to fix. We know the pathways, the major ones that control those. So that's the foundation. And now we're coming in with the ability to look at billions, if not trillions, of chemicals and gene combinations to be able to affect those systems and not just slow down one hallmark of aging, but hopefully slow them all down. And then I hope in 20 years we'll be able to reverse that as well. But if I was, Andrea, a young person your age, I would do a whole lot of genetics, bioinformatics, pharmacology, get a broad education, but definitely learn how to code, because that's going to be essential.
Great. Thank you so much, David. I think we're going to have to stop here because we have a bus waiting for some people, but really appreciate that you're here. It's been an amazing talk, and hope to see you back next year also.
Thank you for having me.